Is the mammoth coming back? Welcome to Erklärwürdig, worth explaining. Can we bring extinct animals back to life through genetic research? There are different approaches to this. Cloning with preserved DNA. This involves inserting the cell nucleus of an intact DNA sample into an egg cell of a living relative. One example is the Pyrenean ibex, which was briefly revived by cloning in 2003, but died after a few minutes. The problem is often that the DNA samples of extinct animals are incomplete and damaged. Another possibility would be to insert the genetic information of an extinct species into the genome of a closely related living animal. Scientists are working on genetic analysis and technologies to create hybrids. The current plan is to integrate the genes of the woolly mammoth, which became extinct about 4,000 years ago, into the genome of the Asian elephant. These new mammoths could live in the tundra and protect the permafrost from thawing through their movements and feeding habits. Permafrost soils perform important functions. Among other things, they store tons of carbon. As long as the soil remains frozen, the release of large amounts of CO2 and methane is delayed, which slows down global warming. It is difficult to understand the exact function of each gene. Even if the genome is completely reconstructed, important information and environmental influences that are important for complete reconstruction are missing. Selective breeding would also be a method to create extinct animal species, at least in a similar way. Through targeted breeding, animals with certain characteristics are selected to resemble an extinct species. Living relatives are used to reconstruct characteristics of the extinct species through backcrossing. One example is the aurochs, an ancestor of modern cattle, which is to be reconstructed through targeted breeding programs. The results are often incomplete. Animals look similar to the extinct species, but are not genetically identical. In addition, the process takes many generations. There are still many challenges to actually reconstructing an extinct animal. DNA decays over time. Most available samples come from fossil remains or frozen specimens, which are often damaged or incomplete. Even well-preserved samples, such as those from permafrost, are only fragments. The quality of the DNA is crucial. Without intact samples, genetic reconstruction is impossible. Even if you have the complete DNA of an animal, it is difficult to create a living being. Genes work in complex networks that are influenced by chemical changes in the DNA and the environment. Without the specific circumstances in which the animal originally lived, it is unlikely that a genetically reconstructed animal will exactly match the behavioral and biological characteristics of the original. A single animal is not enough to revive a species. A population with genetic diversity is needed, which is difficult to achieve with current methods. Resurrected species may have difficulty surviving in current ecosystems. For example, their food sources or habitats may have changed. There is also a risk that they will cause new problems, such as competition with existing species. Protecting living species is often more cost-effective and has a direct impact on biodiversity. The long-term impact of reviving a species on the environment is difficult to predict. Technological advances appear promising. However, there are significant biological, ethical, and environmental challenges and concerns. However, these technologies could also provide valuable insights into the protection of threatened species.